right. Well, yeah. Thanks, Moses, for the introduction. Again, my name is Javier Flores. I'm a data scientist here at PNNL's Earth and Biological Sciences Directorate. Um, Today, as you're seeing on the screen, we'll be talking about the foundations of AI and ML in biology. And I thought there's no better way to start a presentation like this than to ask AI itself uh, what it would say that it is, right? And so we have here at PNNL what's called the AI incubator, which is our own kind of internal version of like ChatGPT. And so I gave two very similar prompts to our AI incubator. They're bolded here at the bottom. The first prompt was draw a picture that represents what artificial intelligence and machine learning is to a non-technical audience. And the second prompt was use a picture to define what artificial intelligence and machine learning is in an easily understandable way, right? So very similar prompts, but we got very different images from each of these, right? And I thought what was particularly interesting about the image on the left is that you have these kind of elements of nature and the natural world involved. And we happen to be talking about foundations of AI, ML, and biology, but nothing in my prompt kind of indicated as much. So if this means that AI is now at the point where it's predicting the future, I'm a little scared. But hopefully it's just, some weird coincidence. But more to what we're seeing in this picture, I think what was the intent of the LLM that generated this was to say that AI can serve as kind of this interface between humans, right, that seem to be in dialogue with this robot representing ML and learning about the natural world, right? This robot is interconnected with the sky and environment around it, right? And presumably it's simulating this information and communicating it to the people that might be interacting with it, right? The picture on the right, on the other hand, is a much more mechanical and perhaps practical view of what AI is, a task-focused view, and that it's showing AI also critically communicating with humans, right? But branching out from AI are all these different gears, light bulbs, data, suggesting some kind of action as far as using information to do stuff, right? And so while definitely not a precise definition, I think both of these images give a good sense of what AI is, and that is something that helps us understand the natural world through using and ingesting data, right? And so if I were to kind of provide maybe a more precise or specific concrete definition, what I'd actually do is partition artificial intelligence from machine learning, because while these two things are typically used interchangeably, there is kind of a distinction between the two. So artificial intelligence is a broader concept that will describe the algorithmic systems that can mimic human intelligence through reasoning, learning, and acting. Whereas machine learning is a very specific kind of artificial intelligence that's oriented towards the autonomous learning from data, typically in this predictive context, right? So can we build a model that, can predict one thing versus the other? Can we learn patterns in our data that might kind of inform reasoning or something that we're interested in learning about? And so if I were to tie these two definitions back to the previous set of images, what we might say is that AI was the part that was responsible for actually interpreting my query, deciding the specific keywords that are important, what action that I wanted to be done, which is the image generation, whereas ML was the component of the AI that was able to know or had learned that given these important keywords, what are some of the images that are associated with these keywords? And then maybe let's give a composite representation of those images to fulfill this request, right? So summing that up, you might say that ML is what drives the learning that empowers the reasoning and decision making capabilities of AI, right? So this talk then is going to be focusing on that machine learning aspect of artificial intelligence, because that really is the driving force for AI to do the things that it can do, right? So we'll start by going over briefly some basic terminology and defining the two types of learning, which are shown here, unsupervised and supervised learning. And then we'll go into common algorithms for each of these and an example application to show kind of how these algorithms have been used. So very basic stuff that we're starting off with. Sample, what do we mean by that? That is the fundamental item or unit of analysis. So I work a lot with GCMS data, and so we might think in that context of a sample being a single sample spectrum, GCMS spectrum. Then features are going to be the distinct traits or characteristics that describe each of our sampled units, right? So continuing with the sample spectrum as our sample, the features might be the different biomolecules that were identified in that spectrum and their corresponding intensities. And so we typically represent features in what's called a feature vector, right? And so if we're talking about numeric values or categorical values, which would be labels, whatever it might be, you'll have just some kind of arrangement, a sequence of values, right? Each corresponding to the different features. And we say that a p-dimensional feature vector, that dimensional piece, talks about the length. How many features that do we actually have? So then data is just going to be that collection of samples and their features. And then if we move into the different types of learning, as you've seen through this cartoon, um, basically we have types divided by the kind of data that we have, right? So unlabeled data, meaning maybe there's some response that we're trying to find patterns based off of, but we don't know what's what, right? So we might be given a collection of data and we don't know that there are viral and non-viral samples in it, for example, right? Exposed, unexposed, but maybe there are some patterns or structures that might hint at that kind of distinction. We just wouldn't know that they would be called viral or not or non-viral. And this is as opposed to labeled data where we have explicit labels for each of our samples where, looking to the cartoon, we know something's an apple or not, right? So depending on, again, the nature of data, that really dictates whether we'd want to consider unsupervised or supervised learning methods. So we'll start with unsupervised learning methods, which themselves can be broken down into two subcategories, distinguished by their analytic goal. So we have dimension reduction or feature extraction methods, which are those that aim to take all of the original features that we have and then find some reduced set. So let's say you had 1,000 different things that you measured on your samples, but that's just way too many for you to analyze meaningfully. Feature extraction or dimension reduction methods will whittle that set down to something like 10 features, right? If you find a really good method. And so the analogy here I thought was kind of interesting would be, an art restoration. Admittedly, this is a very bad art restoration, but it gets at kind of what happens with these dimension reduction techniques, right? Because you might start with something, your original data that's very detailed, very nuanced, has all of these aspects to it. But after dimension reduction, you're going to lose a lot of that information, right? You're still going to keep the main bits. You're still going to keep kind of the main idea of what that original data looks like, but that's going to be kind of limited because you're getting this simplified representation. So this might be a case where you're not using a very good feature extraction or dimension reduction technique. There's definitely a balance between information loss and simplification of your data, and that balance wasn't hit too well here, clearly. Um, Clustering, on the other hand, a lot more straightforward. I didn't really need a picture for this because I'm sure most people understand what I mean by clustering, but I thought it was a cool little animation, smiley face, happy, you know. But this algorithm that's doing this right here is one that we'll end up talking about a little bit later on. called DB scan. But clustering is, as you would think, we're grouping points based off of their proximity to one another. So between these two categories, we'll start first with dimension reduction. And perhaps the most famous dimension reduction algorithm is PCA, principal components analysis. And what PCA is basically doing is just taking your original data and reorganizing it into what are called principal components. The way that it reorganizes the data, though, are according to two main conditions. The first is that you want to find a principal component that maximizes the spread of your data, right? So if we're looking at this picture right here, our data points are most spread about this direction. And we see this rotating line here. This represents our principal component. The reason why we want to orient along this direction of greatest variability is because the errors, which are these red lines, are minimized. Their length is smallest when you're along that direction, right? And so PCA finds this new coordinate system, this new axis to rotate your data onto so that you capture the most variability or information in your data and you're minimizing that reconstruction error, okay? And similar to PCA, there's another popular technique called principal coordinate analysis. And the only difference, or one of the main differences rather, between PCOA and PCA is that there's a step that precedes this rotation, this transformation, where we compute a similarity or distance matrix between our points. So all that this really has an effect on doing is changing the consideration of variability to a consideration of distance. In fact, we could even think of PCA as a special case of PCOA, where PCA uses a Euclidean distance metric, but PCAOA has that flexibility and has that interpretation of preserving or prioritizing, capturing the spatial relationships between your data as opposed to this notion of variability. And then the last technique in these feature extraction algorithms that I wanted to share with you all is T-distributed stochastic neighbor embedding, which is definitely a mouthful. We typically just call it TISNE. And this takes a very different approach from the previous two in that we use a probability distribution to kind of model how close points are or how similar they are to one another in their original space. And then to get some reduced representation, we'll first pick a lower dimensional space. So let's say we had 100 features. We wanted to reduce it down to 2 features so we could visualize that on a 2D grid. In that 2D space, we find an approximating distribution to that original one. And then given that approximating distribution, we can map onto that lower dimensional space. The reason why TISNE is popular is because it allows for non-linearities in your data, right? So PCA, PCOA, they both assume these kinds of linear structures, right? Where you have these very clear straight axes, that you could project your data onto. But TISNE allows for these really weird kind of swirled nonlinear type structures. You could imagine like if we were to take the same kind of axis and try to rotate it on this swirl, for example, the PCA wouldn't know what direction is greatest in variability. And even if it did, really wouldn't capture a lot of information because there's several other candidates that are equally spread out. So you're not going to do a good job in capturing this information. So these tables here are kind of picking out some of these important considerations, some of the things that are assumed by each of these methods, when it might be useful to use them. I'm including it here more for reference, less for me to actually go through each of these points. But the biggest takeaway that I wanted to leave with you all for these algorithms in particular are that PCOA is kind of the in-between of PCA and TISNE. It's a more flexible version of PCA. PCA is a special case of it, but both PCA and PCOA, do assume some amount of linearity, whereas TISNE does not. But where PCA and PCOA have an advantage over TISNE is that they're deterministic methods, meaning that you could run them on the same data over and over, and you're going to get the same result. But TISNE, this isn't necessarily the case because we're dealing with probability distributions and approximating probability distributions. Even running it on the same data, you might get something different. And so it's always good practice to kind of do that repeated runs with different initializations to make sure that your clustering or your visualizations of your data remain fairly consistent, right, and robust. So if we're to move on then to some common clustering algorithms, we have k-means to start. And this is a very intuitive method in that we're just partitioning our data into different clusters with the goal of minimizing the spread of data within a given cluster, right? So we could see that on the picture on the right here, where we might start with clusters that all of these blue points, very variable, all of these red points, a lot of spread, a lot of variability. But as the algorithm completes, the final cluster assignments have much less variability. The spread is less pronounced in each cluster relative to where it started, right? So you have these kind of spherical groups of points that are separated from one another, and you know, very intuitive as far as what we're trying to do with k-means. Though here we do have to choose choose the number of clusters, which is something that can be guided if we were to look to validation metrics like a gap statistic. There's several others. These are all empirical measures, right? Data-driven measures that help us, okay, given our data, what does it seem like there are as far as the number of clusters? Next, we have agglomerative or divisive hierarchical clustering, where also very intuitive, we're either iteratively grouping up data points, starting from them being each their own cluster to at the other extreme, all being grouped together. That would be agglomerative clustering, and divisive would be operating in the opposite direction. And what kind of dictates the points that are joined together or the points that are separated is going to be a similarity metric or a distance metric and a linkage method, right? And so depending on the choices of these metrics and methods, you might have different points grouping together, different collections, you know, forming out different clusters forming. And then last, the smiley face algorithm, DB-scan, density-based spatial clustering of applications with noise. This is the most flexible of the three clustering methods shown here. You don't have to specify a number of clusters in your data. This algorithm will figure that out automatically. But what you do need to specify is a search radius, right? So how far outside of a given point to look, and then a minimum number of points that you're looking for in that search radius. And so the way that this algorithm will work is it'll go through each point, draw out that radius, and then count how many points are in that radius. So here, if we say minimum points of 5, and we're looking at this red point, we see that, hey, there are 5 points in this radius, so this is going to be a core point that's clustered. Moving on to another point, we see that also has that condition, and crucially, it shares a common point with that prior core point. So then these all would be clustered together, right? And so that'll continue until you don't find points that meet that criteria. And then if there's no common points, you have distinct clusters. These gray areas, or these gray points rather, are those that didn't meet any of these conditions. And so they would just be labeled as outliers, right? So whereas k-means and agglomerative or divisive hierarchical clustering might be really affected by outlying points, DB-scan is insensitive to them. They're not going to be thrown off. They'll just say, hey, I don't know what to do at this point, so I'm just going to call it an outlier. It doesn't seem to be grouped with anything. So again, tables for you all's reference, but the key thing here is k-means is probably the most intuitive, straightforward, but it does require that you choose k, you have a certain shape of cluster in your data. DB-scan, on the other hand, is probably among the most flexible, but you really do need to be careful in how you define your search radius and the minimum number of points, because you could imagine a case where, let's say you specify too large of a search radius, then everything's going to cluster together. Or if you have too few number of points, the same kind of result would happen. So there's a more deliberate tuning process that's involved with dbScan relative to k-means. And then hierarchical clustering typically is employed when you expect some kind of nested structure in your data. So how have I used these different things in a biological or chemical context? Um, the example I'm sharing here is from my own work, done with others here at the lab, um, relating to matching or annotating compounds in GCMS spectra, right? So as some of you may know, the way that we identify these compounds in spectra is through using similarity scores. So we'll have our sample spectra, and we want to match it against spectra and reference libraries, quantifying that match using a similarity score. And the problem, though, is that there's several scores, right? And there's not a real clear consensus rather on which of these scores is best. And so this gives rise to analytic uncertainty, reproducibility issues, which in turn can lead to inconsistent annotations, right? So somebody might choose or think a better metric should be used over the one that I was using, say, that could lead to differences in what we say the match is. So the question that we were faced with is, well, can we group these metrics in some ways using historical data, right? Are there clusters of similarly performing similarity score metrics that we could, reliably say that they're going to lead to the same annotations? And then a step further than that would be, given these clusters, assuming they exist, are there some clusters of metric that perform better than others, right? And so the answer to both questions is yes. And the way we found this out was using agglomerative hierarchical clustering. So there was actually 66 different metrics that we considered. We computed them on, historical spectral data, and then their computed values were correlated with one another to form this heat map here, right? So we have each of these metrics along the axes and their correlations kind of visualized in this heat map: red, highly correlated; blue, highly anti-correlated; yellow, not correlated at all. There were clearly 4 clusters that came out of this, and we found that cluster groups 1 and 2 specifically tended to better separate the true match relative to others. And so fortunately, this all told us that the majority of metrics that one might consider are largely highly correlated with one another and perform well, right? So that allowed us to, breathe a collective sigh of relief, not worrying that, there was some major reproducibility crisis on our hands with GCMS annotations. So that's unsupervised learning in a nutshell. I mean, the intent of this talk is to give kind of an overview of some of these different methods. And so we'll next go into supervised learning, which, similar to unsupervised learning, can be broken down into 2 subcategories. Here, those are regression, which are where we're modeling some quantitative response, or classification, where we're modeling a categorical response, where we have different categories. And so I also used AI Incubator to generate, some images here, just because I felt like we needed something to spice things up. And AI Incubator did not disappoint, because it gave us a human cat hybrid and a human book hybrid in a classroom, which was really interesting to see, if not slightly disturbing. But there you are. So regression algorithms. We'll start with partial least squares regression, which is very much related to PCA, in that, like PCA, we're trying to find kind of a rotation or a transformation of our data that allows us to maximize something. In this case, what we're trying to maximize is the covariance between our data and the response we're trying to predict, right? So we can understand this better by looking at this picture. we're showing the PCA component. So this would be what PCA would say is the first principal component. Why? Because this is where the data are spread out most. And we could contrast this with the partial least squares component, which is in a totally different direction, but crucially, it's in the direction where we see a clear gradient in Y, right? Y goes from a low value to high value in this direction, as does our data go in some amount of variability. So PLS is going to differ from PCA in this regard. It'll find that direction that maximizes the covariability between our response and our data. And so it's seen as kind of a blend between PCA and regression, because given these PLS components, we'll throw them into a regression model to actually get predictions. Gradient boosted trees, on the other hand, operate entirely different, have no relation to PCA. This is what's called an ensemble method, which is just a collection of smaller models. In this case, the smaller models are decision trees. And what decision trees are just branching partitions of our data, right? Branching decisions that you make given the values in your data. And for gradient boosted trees, what we're trying to do is iteratively minimize the prediction error based off of prior trees. So as shown in this picture here, we might start with a first tree fit to our data. It has some amount of prediction error. Those errors are then used to train another decision tree, right? And then collectively, those will still have some error, and so on and so forth until you have, perfect prediction of your data or good prediction of your data. Because this process works the way it does and that we're iteratively improving upon the errors of the last model, overfitting is very much a concern where we could get to the point of perfectly predicting on our training set. And so we often try to control this through learning rate specification and feature sample subsampling, because overfitting would mean that we could only predict well the data that we have and not kind of new data. It wouldn't be a generalizable model, so it'd be functionally useless. And speaking of overfitting, a very common model for this problem are neural networks. And this is because as flexible as this modeling paradigm is, it requires a lot of data so as not to be too good at predicting its training data, right? And there is a specific property for neural networks that something about like universal approximation. So any kind of functional form that might truly exist, a neural network can approximate it, right? And so you could see how this kind of property lends itself to overfitting. You might have all these nuances in your training data, some that correspond to true signal, others that don't, but the neural network can learn all of that. So there's a very clear danger of overfitting here, but more to how this works or what it is, we just have this interconnected layers of artificial neurons. The reason why we call them artificial neurons is because they take inputs, they transform those inputs, and that transformation usually is subject to some activation threshold where some value needs to be met to have it output a non-zero value, similar to biological neurons that have this action potential firing mechanism. So things to look out for, tables, again, for your reference. And the biggest thing to point out here, again, is overfitting as a concern for gradient boosted trees and neural networks in particular, more so for neural networks, just because of their flexibility. Neural networks also need a lot of data to work well, whereas gradient boosted trees and partial least squares can operate with more moderately sized data. So home stretch here with classification algorithms, we have discriminant analysis, which again is like PCA in that we're finding a new transformation of our data. In this case, we call those discriminant functions. Those are the things that transform our data. But what we're trying to do is find transformations so that the clusters of our data, the categories, are separated from one another. And then within categories, we have a clustering of points, right? And so given this projection from these discriminant functions, we could throw new data through it, and then wherever those new data land in that new space, If they're closer to, say, the red points, they'll be grouped as a red observation. If they're closer to blue, they'll be classified as a blue observation. Random forests are another ensemble method, similar to gradient boosted trees, but they differ in that rather than sequentially training decision trees, they'll do this in parallel, right? So we have a lot of independently grown trees that might use different samples, different features in growing them. And then we get a prediction from this model by a majority vote situation, where, if 60% of these individual trees said that a point was supposed to be red, for example, that would be the classification. And then last, we have support vector machines that are probably the most dense in how they work among all of these, but at a high level, what they're trying to do is find a decision boundary to separate points. So if we look at this figure on the bottom right, we see that if we were to just draw a line here, there's a very clear separation between blue and red points. But you could imagine a situation where these points are more interspersed among one another, and so a straight line wouldn't do well. And so support vector machines are nice because you have something called a kernel function that allows you to draw more curved and wiggly kinds of borders, right? So you could get around this interlock nature and still kind of cleanly separate your two sets of points. And so last set of tables for you all, but what I'd say is about these and the previous set of supervised learning algorithms is, even though I've classified them as either classification or regression, really most of these can be used in either case. It was more of an artificial distinction that I used here. And I'd also say that the random forest algorithm in particular probably gives you the most bang for your buck, in that you have a lot of flexibility in the kinds of relationships you could model in your data, similar to a neural network, but you don't have nearly the same amount of sample size requirements. So this is often a go-to algorithm when you're looking for something that's interpretable, doesn't require a lot of data, and is still highly flexible. So how I've used these algorithms before is also in a spectroscopy type example, but rather than GCMS, NMR spectroscopy. And the problem here is also relating to identifying kind of which compounds are in these spectra, because NMR profiling, which would be including identification and quantification, is a notoriously low throughput process. And so we were trying to find a way to automate some aspects of that. And given kind of the reproducibility of spectra, in that we see kind of the same peaks corresponding to these different compounds across NMR spectra, the thinking was, well, let's use historic NMR spectra to build models that could predict presence, absence of different compounds. And so we built this system based off of a collection of different classifier models, sorry, that did just that, predict presence, absence of metabolites based off of historic data at EMSL. And the models that we considered included random forests, neural networks, support vector machines, everything that we've talked about on these previous slides. And what you're seeing on the right here is just kind of high level accuracy of this recommender system in identifying presence absence based off of this evaluation data set of 585 spectra. So accuracy above 75%, which is really good. This table on the bottom is a snapshot from the tool where we have this implemented. So NMR analysis is that tool. And what you're seeing are metabolite names, their predicted probability of actually being in the given sample spectrum, the model used, and some evaluation metrics for how well that model had been found to do. So wrapping it all up, I know kind of a whirlwind going through each of these things. AI and ML, highly interconnected concepts, but ML is really the means for AI to develop its intelligence. There's a lot of different categories of ML methods, really driven by both your analytic goals and the kind of data that you have. And if the two examples that I've shared aren't enough to kind of show the power of them to biological research, what you've seen through Lisa's talk and what you'll see through the rest of the week certainly will show you this. And then I'd want to end on just another plug for some other cool tool that EMSL has built, which is the slope application. This is built specifically for people who may not have or experience with machine learning or comfort with it. And so the total novice that knows nothing can provide their data to slope and be walked through um, an analysis to implement machine learning, depending on their goals and the data that they have. And with that, I will take any questions.